So, DNA sequencing. What is it and what is it used for? I'm Leslie, and this is my cat Callie, and I'm going to be explaining everything you need to know about DNA sequencing. So again, what is it? By definition, it is the process of determining the sequence of nucleotides in a piece of DNA. The most commonly known process is called Sanger sequencing. At first, I thought it was this really difficult thing to understand, but it actually turned out to be really easy. The process of Sanger sequencing includes these key components: template DNA, nucleotides, DNA polymerase, a primer, and fluorescently labeled dideoxynucleotides. First, the DNA is denatured, meaning that it changes from double-stranded to single-stranded, so the primer can easily attach to it. DNA polymerase then lengthens the newly made strand until it randomly places a dideoxynucleotide. This dideoxynucleotide has a missing hydroxyl group. The hydroxyl group allows for nucleotides to bind to one another. It missing means that the chain has become terminated. It should also be noted that these terminations have an almost fluorescent color to them. Multiple chains are created, each with their own dideoxynucleotide at the end. With these strands, they are then assorted by length. The fluorescence, each color associated with a particular nucleotide, will reveal the final sequence of the DNA. So now we must ask, why is DNA sequencing important when studying evolution? DNA sequencing is often used in evolutionary studies to answer questions such as where we came from, where we evolved from, and how it correlates to genes and traits. DNA sequencing is able to track changes in genetic code over time. By sequencing the genomes of different organisms, we can identify which genes are possible for which traits and how those genes have changed over the course of evolution. We still have one question left to answer. What are some modern-day uses of DNA sequencing? DNA sequencing is used to obtain detailed information about the genetic makeup of living organisms. It is also used to identify genetic variations associated with diseases, which helps in the development of new therapeutic tools and medicines. Through DNA sequencing, we have also found out that we are almost 99% related to the chimpanzees. So, I didn't know this, but apparently we're closely related to mushrooms and bananas. So that's interesting. Bye.